I have to unmute. Everybody hears me? Yeah? I also would like to thank the organizers for inviting me here to this fantastic symposium. And Peter said this morning that Nikos was a hero for him. The same thing holds true for me, but he is also an example and an inspiration. You know, I think he has been an inspiration for many people. And I don't know whether Brian is here. I'm not a laid-back Californian, so I will hug him actually. Okay, having said that, we have been more or less in the same business. Is it okay? Yeah. For quite some time. Starting to do fMRI about 20 years ago, and I remember the very first time that we gave actually talks at Neuroscience, where Nikos, me, and people from Tom Albright's lab were presenting imaging. And before that, in another life, basically, and I was hoping for a cloudy, rainy afternoon because you will see nothing of my slides, definitely not the first one. Well, I also want to thank first all the people that were doing the work that I was presenting, that I will be presenting. Two Chinese colleagues of mine, a PhD student and a postdoc who just left the lab. Chi Zhu is now at Neurospin near Paris, and Xiaolian Li, who is still in my lab. Really great, brilliant Chinese students I worked with over the past couple of years. So to come back to my early life, and yeah, you don't see anything, but you have to believe what I'm saying. These are flattened sections through the monkey brain in which we had done 2-deoxyglucose experiments. We simply labeled metabolic activity in the brains of these animals, and what was apparent in these sections basically were like many columnar structures all over the place. Striate cortex, extrastriate cortex, parietal cortex, infratemporal cortex, prefrontal cortex, all over the place. The only thing is, we couldn't make any sense of it, because we could only label one condition at a time, sometimes two conditions at a time, most of the time no difference in metabolic activity. And this basically inspired me to get into a technology that would allow us to measure these kind of small mesoscale structures in vivo, and not only at the superficial part of the cortex, which is accessible with all kinds of imaging techniques, to photon imaging, etc., but also buried into soul cell. A holy grail, actually, for many people doing imaging. I think Brian will confirm that happily. So, of course, this is another one. Of course, you could turn to technology that was in the hands of people like Nikos, quite expensive equipment to try to do these kind of things, and Nikos achieved that very well, and I'm sure we will see quite a few examples later on. But our resources didn't go that far, and so we had to return to sort of like the poor man's solution to solve that problem. And the poor man's solution was also like poor grad student, postdoc life, getting into the hospital with monkeys basically after hours, which meant starting working on hospital scanners, regular hospital scanners, late at night, typically after 8 p.m. Another poor man's solution is that we relied, oh, the image is really bad, on rusted iron. This was just a piece of rusted iron, and also some copper wires. So the combination of these kind of poor man's solution technologies helped us basically to get not even to the knees, but to the ankles of Nikos. And I hope I can convince you, at least if the projection system allows me. So what am I gonna talk about? I will talk about technology that we developed in our lab in collaboration with people in Boston at MGH. in particular implanted coils, phase array coils that are embedded in the headset of animals. And I will show you some examples. And I will show you that we use the technology in a system where we knew that we should find sort of like small-scale functional modules in the brain, in particular area V2. We have heard several times about V2, where there are compartments which should be separable when you use the adequate stimuli. I will also talk about using this technology to have a finer-grained view on the topographic, and specifically the written topic organization in visual cortex, and also at these columns that we start seeing now all over the place, from occipital way up to prefrontal cortex. Category selective structures, motion selective structures, and finally, which I think is very important also for non-human primate imaging, going to cognitive tasks, and then trying to see what's really going on at brain scale, network level, but also at very fine local single unit level, etc., for doing things that you're not capable of doing in normal human subjects. All right. So what we do is we take CTs of these animals, and then for example, based upon probabilistic maps that we acquired in previous animals, and this is sort of like one slice through a probabilistic written topic map based upon more than a dozen other animals, getting that into the space of the animal that we still have to implant, which has not been used yet, getting that into the space of the animal, and then you can sort of like use all kinds of software to guide where you want to go with electrodes, injection needles, to do optogenetics, etc., even before you have done any experiment. And then you build everything using the CT skull, and this is just an example of an animal that we implanted. You see these copper wires, and then you can embed up to 4 or 5 recording wells simultaneously with these coils. This is what we do, and basically this is an example of a monkey where this has been done with 4 different recording wells, and in this case an 8-channel coil embedded. Nowadays we go up to 10 channels, which is beneficial for imaging because you can image faster, you can do multi-band imaging, so you get like better quality of data. It also works pretty well. Actually, the very first animal that we ever implanted with such a device is still alive, and this was implanted more than 9 years ago. And it has pretty good coverage. So this is a study on that first animal, basically showing the profound increase in sensitivity here with, in that case, we used an 8-channel implant, phase array implant coil, relative what most other people, except for Nikos, are using, like external devices, external whatever phase array coils. So you see here in red, you get like a significant increase in sensitivity. So basically, nowadays, together with these higher count phase array coils, we get an improvement of about a log unit, which means that we can scan the brain either with higher sensitivity, keeping the resolution the same, but having more sensitivity if you're looking at subtle differences in functional properties, subtle differences between tasks or stimuli or things like that, or you can buy that sensitivity to get to higher resolution. Yeah? And basically, instead of what we used to use in the past, like around 1 millimeter isotropic voxels throughout the whole brain, we are now going to 0.5, typically 0.6 millimeter isotropic voxels. So this really is beneficial in many ways. Also, once this coil is there, it's always at the same place. You get always the same kind of loading of these coils, and so forth and so forth. And little can go wrong. It's all embedded in the head post of the animal. Of course, the electronics attached to it, that can go wrong, and that requires maintenance. So for the rest, we use regular scanners, as I said before. We plug in monkeys, and we train monkeys to do all kinds of tasks, usually fixation tasks, but always also with hand movement control to make sure that these animals are really sitting still, which really helps a lot in terms of the quality of the data. Contrary to what Nikos usually does, as you can see, of course, our animals are sitting in a horizontal position, while Nikos has a vertical, well, and some other people as well, have a vertical bar, and the monkeys are sitting in a system which is typically used in most awake behaving monkey laboratories. And I know Nikos was critical about that approach many years ago, and I hope I can convince him that it does work. You can do quite a bit of things actually using this approach. And we use also, that's where the rusted iron comes from, we use contrast agents. So we can inject contrast agents, and the sensitivity also increases again compared to regular bold imaging. We have now also adapted the whole system to get actually very wide field visual projection capabilities up to more than 100 degrees in diameter. So we can really look at big portions of the visual field to be stimulated. So proof of principle, does this work? Easy a place to start with is actually, well, easy. Quite convenient place to start with is area V2, which is characterized by stripes, thick, thin, and interstripes. And this is a section I also acquired in my previous life. This was cut, again, parallel to the cortical surface in area V2. And if you stain that for cytochrome oxidase, you can recognize these stripes. You see, well, I hope it's good enough in the back of the room. You can recognize thick stripes, you can recognize interstripes, and thin stripes, and thick stripes in between. Now, it is known from many studies, electrophysiology, also optical imaging, that different types of stimuli will activate these stripes in a different way. For example, thin stripes, the thin stripes here, this is again that deoxyglucose experiment or experiments that we have been performing. Thin stripes, they are preferentially activated by colorful stimuli when you compare that to achromatic stimuli. So there is a bias for color selective, or there are more color selective cells in the thin stripes compared to the other stripe compartments, which doesn't mean that there are no color selective cells in the other stripes, but there is definitely a bias. Also, that's known from many other studies, the thick stripes, there is a bias for disparity signals, depth signals, and also for motion, what Semi was talking about this morning. So there are a couple of functional markers which are easy to use to see whether you can activate these different stripe patterns with fMRI. So another thing is difference in myelin, and a proxy method to look at differences in myelin is sort of like taking the ratio between T1 images, and Brian also mentioned that, and T2 images, sort of like it gives you an approximation of myelin density. And there are indications, and even the histology literature is a little bit mixed there, that you can differentiate actually the pale stripes from the other stripes based upon a myelin content. So we thought, okay, maybe we should try to use this T1, T2 ratio to see whether we get like a differential stripe-like pattern using that measure with MRI. There have been people, including Brian actually, using human subjects on a high-field scanner that were able to visualize these different stripes, or some of these different stripes. I just put up something from the lab of Roger Tutel, who found here, as you can see, like stripe-like patterns using or in human subjects. So, challenge though is that these stripes in the human are a bit wider and more spread from each other than in a monkey. So it is a bit more tricky in a monkey than to do it in a human. This is the way how we try to visualize these myelin contents, difference between T1 and T2 contrast. So what did we do? We had monkeys implanted with these phase array coils, putting in a scanner, and then they were trained to fixate. They're fixating here, and I'm not sure it's very visible, but there was like a colorful circular grating that was moving, contracting and expanding. Monkey kept fixating, then there was nothing, and then there was sort of like an achromatic grating as well, and the monkey kept fixating. In the other experiment, we did exactly the same, but now we showed patterns which were defined based upon disparity. This was like a moving disparity stimulus, a big moving disparity stimulus compared to the monocular controls. And this is basically the summary of that story. Here, is that visible in the back? In green, you get like disparity selective, or maybe here it's easier to see, green disparity selective stripes within rhythmotopically defined area V2 with in the black outline. We had color activations with interdigitated with these disparity stripe-like or biased patterns. And in the activity map, just like a heat map, we show the myelin density values. And we observed, if you do sort of like quantification, we observed that these color and disparity biased stripes basically had different, significantly different myelin content in these compartments. So in the follow-up study, this was published actually, I think, last year. In a follow-up study, we also added a motion localizer, a very big motion localizer with this large field of view motion, comparing just moving versus static patterns. And there you see more or less the same thing. Here is again in blue the color stripes, and in yellow activity for motion. So again, also with motions, you see this nice segregation between the different stripe patterns in area V2. More or less the same what people have been observing in optical imaging and with electrophysiology. So this gives us a little bit of confidence that even at 3T, you can start segregating these like relatively fine-grained patterns. And here is disparity. You see disparity, and motion basically gets into the same subcompartments of area V2. So we see thin color-biased stripes, thick disparity and motion-biased stripes, and then also we have some evidence. It's not as good as with the functional markers of different myelin interstripes compared to the thick and the thin stripes. So it is possible, yeah? But you learn nothing new with it. It's just like to confirm whether or not this thing is working. So then we went on to different types of experiments where we also know a lot about the system, but maybe not necessarily the details, the nitty-gritty details. And we started running basic rhythmotopic experiments, exactly the same what Brian was talking about, using phase-encoded rhythmotopic mapping techniques, sort of like wedges that are rotating, or annuli that are expanding and contracting while the subjects are fixating in the middle. Exactly the same as what Brian did in plenty of human subjects. I have to give a little bit of background for people that are not really familiar with it. The textbook idea, and actually this was sort of like what was imprinted in my own brain, because this is what you learned at school and how you grew up with, is that you have like area V1, and then you have like slabs of area V2, and V3, which are parallel bands, very nice parallel bands, when you move in more rostral directions relative to area V1. That's sort of like the typical monkey model. Higher up, area V4, some authors, and actually quite a few authors these days, they say that this V4-ish area can be broken up in multiple subcompartments. Actually, Semir was the first one. Is he still around? Yeah, he was actually the first one to suggest that. In New World monkeys, in marmosets, and in owl monkeys, which do have like a flat cortex, basically, the situation is a bit more complex compared to these old world macaque monkeys. In New World monkeys, quite often you have like multiple upper field, here these pluses, upper field representations embedded in, we're talking about dorsal cortex, in typical lower field representations. Much more complex than in the schedule of the macaque monkey. Based upon these New World data, basically, people like Rossa, Marcelo Rossa, he sort of like also put up alternative old world schematics, where you see that this V3 thing, there's this very small parallel band anterior to V2 starts to be broken up in several subdivisions. So it's quite confusing, right? So we set out, and actually we, in earlier studies, we sort of like adhered to this model. So it was really the way we were thinking, and we thought this is sort of like how it is, right? In some studies here, like in the review paper schematized, we see these parallel bands of V1, V2, V3, etc. Yeah? Also by other studies. So then we turn to this high resolution imaging, and when my students with 0.6 millimeter isotropic voxels all over the place, like we scan the whole brain at this resolution, and it's very similar to what also again Brian, so it was a great talk because he basically introduced whatever I'm talking about. We use rich stimuli. We use sort of like not stupid or not only stupid checkerboards, but things that the visual system is interested in, moving faces, walkers, etc., etc. And the monkey fixates, and then these things are rotating, or you have annuli which grow from small to far periphery. Wow, is this visible? This is raw data. This is literally raw data, unthresholded and things like that, how it comes, well, after getting the faces out of these responses. And just to make it a little bit easier to understand, red here, these little stripes is the upper vertical meridian, green here is the lower vertical meridian, and blue in between is a representation of the horizontal meridian. Of course, we have everything in between, but this is just to simplify it. This is very noisy up here, and you see in early visual cortex, this is V1, V2, V3. In early visual cortex, you get this stripe-like zebra type of pattern, which also Brian showed you earlier on in humans. But then when you get to, this is with the 12-degree radius stimulus, when you get into the more peripheral representations, it becomes quite noisy. This is from the same animal, but now with 50-degree radius stimuli. And I hope I'm going to go up and down a little bit. And you see these green, look at here, you see these green stripes and the red stripes here going all the way out to the far periphery of these early visual areas. I keep, whoops, I see that. You see here in the back. Now, this was with a stimulus where we had like moving faces, like really dynamic faces in also embedded checkerboard pattern to have good stimulation of most of the cells. We did exactly the same. We thought that, okay, maybe here this is towards parietal cortex, that we could get better activation when we had like depth varying stimuli, because we know that there is quite a bit of depth selectivity in parietal cortex. We did exactly the same experiment with dynamic depth stimulus, completely different stimulus. And you see, I'm toggling here between these two different data sets in the same animal, except for the crab here, which is probably noise. It's very, very, very similar. I also want to point out here, this is dorsal cortex, red. It's not very visible. Maybe, do you see it, Nikos? Several small patches of upper field representations in red there in dorsal visual cortex. And again, I'm toggling between the two data sets. You see them basically coming back in dorsal visual cortex. So we have like lower field representation and several, not one. Typically V3A has been considered having an upper field representation. We see multiple of these upper visual field representations coming back over and over again, independent of the stimulus that we use, and also in different subjects. Again, something which is very difficult to reconstruct from the existing literature. So these patterns, of course, these color things are nice to look at and so forth, but they're not really very well diagnostic. So what we started to do is to draw the fine eccentricity and polar angle lines upon flattened representations of the cortex. And that's shown here, again, not really very visible because of the light here. But here you have like polar angle lines, and here you have like eccentricity lines of this part of the cortex, dorsal part of the monkey. This is the field sign map. If you overlay, and in dark is the sulcus, this is sort of like this sulcus that you're seeing here, the lunate sulcus. If you overlay the field sign map on top of these eccentricity lines and polar angle lines, you start to, or when the image would be good, you would be able to start to see very fine details of that cortex. And we consistently were able, in I think now 6 animals, 12 hemispheres, to see very nicely this V1, V2 border, where you go from a mirror to a non-mirror field sign map. And then you have V2, and then in front of V2, we consistently started to see like multiple areas, not one area, but very similar as in the new monkey, different representations with several upper field representations, not just restricted to the typical upper field representation in area V3A. We also did some other tricks, exactly as what Brian showed earlier on, calculating population receptive field sizes in these different segments to make sense of it. How can you combine these different quadrant representations, basically, based upon not only the eccentricity and polar angle, is going down, also based upon population. Is there another? Yeah, sometimes it works, sometimes it doesn't. Anyway, we get consistently different quadrants, and using a combination of this information, we can link it to different areas, not just area V3. And I don't have time to get into the details, but we get sort of like a very similar view on this part of the cortex in the macaque as what has been described actually by some people in New World monkeys. So this is sort of like the summary figure of that. We have like a blue sector. This is what we found in our data, a blue sector with the lower field of area V4, and then an area which we think corresponds best with DLP of Marty Serino that he found in the owl monkey. And then we have like a bunch of upper field representations which belongs to the upper field representation of this DLP and also V3A, and then only V3, a V3-like area starts in more parietal directions. Very similar, actually, to what people described in both marmosets and owl monkeys, at least if you look at the rough topographic organization of this. This was with the small stimuli, with the 12.5-degree radius stimuli. The big stimuli that I also showed you, the color maps, there you start to see that V3 gets all the way from, actually this DLP and V3A gets all the way into the parieto-occipital towards parietal cortex. Also in that large field experiment, we did other functional localizers, and that's shown here. Here you see the boundaries of the areas, V1, V2, V3, V3D, and then V3A, and this DLP, the area that we called a DLP. And this is just a motion localizer, and you see how surprisingly well it is confined to V2 and V3, and does not activate V3A, actually something that we described already quite a few years ago, which is different from what people have seen in human V3A. So, I think we have to sort of like revise a little bit what, at least for me, was textbook knowledge of macaque dorsal visual cortex, where V3D is reduced in size. You have an area V3A, and then sort of like a fourth-tier area, which has like a population receptive field sizes comparable to area V4, which we think is a homologue of DLP of the owl monkey. Which also means that at the end of the day, it is more similar compared to traditional views between the owl, between the new and the old world monkeys. And maybe if you start looking in detail in the human, it might be the same. Dorsal V3 is definitely different from ventral V3. There are some maps. Actually, this is an old map from, again, not very visible here, from Sereno, one of the papers also mentioned earlier on, where you see this gap in the field side map. Also, the human connectome data shows similar difference between dorsal V3 in the human compared to ventral V3. Okay, you have to believe me, in the previous thing, it's very detailed analysis. What about things that we don't know about? Yeah, I've shown you the V2 stripes, I've shown you some retinopathy, but I think this is the most fun part. It's new, it's basically from the last couple of weeks. Higher order cortex. We know that there are a bunch of different areas, about 30 in a monkey, I don't know, I heard about 20 in the human, maybe more. And we also know that part of that cortex is category selective. We have the face areas, we have the body areas, we have the scene areas, we have the word form area in the human, probably not in the monkey. But these areas are considered relatively large and homogeneous. Yeah? For example, Dorsao found this area, a face patch or face area in the monkey, one of the 6 typical face areas in the monkey ML, which contains virtually all face selective cells. And it's relatively large. Is this the case, right? So we started doing, and we also found that for the body patches, only a few relatively big areas concerned by processing body information compared to other object information. This is a slide from Doris. You see these relatively large areas specialized in processing facial information. So we started to map them out at high resolution with very well controlled stimuli, 10 different categories, you see them here, and very well controlled, which we also have been using quite a bit in electrophysiology. And then when you do that, we start to see, and I hope this is visible, we start to see much finer grained patterns. Here you see like a face, no, sorry, this is body patches. Here you see like two little guys there. You see like in parietal cortex, in EPL, little dots, yeah? Not very convincing if I just show you this. If I show you, this is day 1, day 2, you see basically in exactly the same locations, you see these small little patches. Here again, you see there, you see here in the depth of the cortex, you see there in the depth of the cortex, occurrence of the same selective patches day after day. This is for bodies. This is a bit bigger. Here you see in parietal cortex these 3 different patches. Here in the STS, remember in the old data there were like just 2 body patches. We see many more of these small functional domains. This is the second day, and you see, I hope it's visible, you see these small guys really coming back over and over again. So it's very reproducible across different days. You could say, okay, maybe it's more sensitive in that region of the cortex, it's activated for everything. No, that's not the case. This is bodies versus objects. If you do that disparity thing that I talked about for the V2 stripes, this is, oops, sorry, this is disparity. This is body from the same animal, and you see these activations basically here also in parietal cortex, mainly jumping up and down. This is disparity day 1, day 2, and again, a very reproducible pattern of activations. Faces, same story. Here, body in blue now, faces in red, and you see this interdigitation of these small different mesoscale patterns in the 2 different categories. One day, other day, and I'm going to point out just small ones that are very reproducible. For example, here, red, blue, red, blue, red, blue, red, blue. So all over the place, basically, this is around in STS, very highly reproducible, fine patterns for faces, relative to other categories or bodies. Also in frontal cortex, I'm going to skip this in the sake of time. Also in frontal cortex we get, and this, here you see these different small patterns, selective in this case for faces, reproducible across different days. And then you can put that on a flat map, very reproducible basically, small scale in parietal cortex, frontal cortex, and with very, very stringent criteria, selection criteria of these voxels. They have to be there in different experiments across different days. But we also have like still rather big patches. For example, that ML, can we look with this kind of technology at differentiation, functional differentiation between these big areas? And that's what we started to do, like looking at functional profiles of individual voxels, clustering of these functional profiles, and also at the connectivity. And for example, this is area ML in the monkey, in the STS of the monkey. You select these voxels upon particular contrast, faces versus control objects. Another experiment, colorful big faces versus control objects. Then you select these voxels, and then you can sort of like plot the functional profile of the individual voxels for these different stimulus categories. Like here, faces, and you see, of course, there's a face patch, so it should activate big time for faces, and less so for others, but there is some activity for body areas. You have all these voxels, you can calculate the Euclidean distance between these voxels, and then do clustering, and then you end up with 3 different clusters within this particular area, which is sort of like the minimal number of clusters of that specific area. So within this ML, you find 3 different categories or different clusters of selectivities. Actually, we have done exactly the same experiment with single units in exactly the same region using exactly the same stimuli. then too we find, based upon single unit responses, three different clusters. So it does fit remarkably well with what you find with single unit recordings. We get this with the achromatic stimuli. You get that with a completely different experiment with these color stimuli, big stimuli, 2-3 times the size of the small guys. Unfortunately, we didn't have like the 10 categories there. But if you correlate the categories that we tested in the 2 stimuli, you get a very high correlation of these functional profiles in very, very different experiments on the same animal. So indicating, if you back map them, that you get like 3 segregated clusters even within this bigger area. We also look at connectivity based upon resting state data in the same areas, completely independent acquired, of course. And you can look, you have 3 clusters in the left and the right hemisphere. You can look at the within cluster functional connectivity and compare it with the across cluster functional connectivity, and that's highly significantly different, which means that clusters belonging or the same clusters are better connected with each other than with the other clusters within that given area, again indicating that these things are probably doing something different compared to this very homogeneous selectivity reported in the literature. You can also shuffle that, and you see that this difference between within and across cluster connectivity is significantly different from what you get by chance. I'm going very quickly now. This is basically the same thing if you go to more stricter selection. And actually you get the same thing in body patches. If you do the same trick in the body patch, you also get like clustering, in this case also in 3 different functionally defined profiles. Yeah? So you get, with this technology, like a better view on what these different voxels really are doing, and in some cases it fits with what we have seen in cells. So we get like a highly reproducible columnar-like pattern all over the place. We saw unpublished patches of the cortex that are selective for bodies and for faces. The connectivity for different clusters is different, which the functional connectivity, which might also indicate that the anatomical connectivity is different. And of course, these are things that have to be looked at in much more detail. At least it gives us a handle on looking at these kind of things in vivo without having to sacrifice these animals. I think I have to. Well, we also see that with motion, basically. We get like columnar organization for flow patterns, very reproducible patterns. I think interesting also even in frontal cortex. Here you see these expansion, contraction, small scale things for big moving patterns. It's not just for category selectivity, also for motion selectivity. I'm skipping the last part. We also applied that in cognitive designs, where we basically do exactly the same attention experiment in monkeys and in humans, a shifting of covered attention. You see SPL in the human activated, you see SPL in the monkey activated, and when you record there, you see shift selective activity in that region. And actually, we only got it because of the high resolution fMRI. We tried to record there, and we were always just above an fMRI patch, and we found one cell. When we moved to the fMRI spot, we found basically half of the cells being shift selective. All right. I think in conclusion, we are getting there, and of course, this is the poor man's solution. If you would marry this poor man's solution with the rich man solution here, I think there are very exciting times ahead of us. Thank you very much.
Thank you very much. We're a bit behind time, but they're going to try and change the projector, which I'm sure is highly appreciated. So we're going to use that time for questions. So, behind you.
precise of receptor fields that were neurophysiologically recorded by Hubel and Wiesel, and also the psychophysical perceptive fields obtained with the Westheimer paradigm, from the fovea to the periphery. And there's very good agreement. But I wonder why in your population receptor fields there's never an inhibitory surround.
That's a very good question.
It's, as Wim says, it's a very good question. In fact, Serge Dumoulin has proposed that he will do a better job fitting the fMRI data with a difference of Gaussian model. We don't always see that. It's right, during the talk that I gave, there was a moment when I said, it remains a rich active field with people experimenting with different models. Kendrick Hayes got the compressive spatial summation, Serge has got the difference of Gaussian, Stelios and George back here have another calculation. So there's a number of these things in play. There isn't a consensus yet. And I think what Wim did and what we did with the first order linear ones is kind of what people start with, but the field remains active.
Any more questions? Okay, in the back.
I had one curiosity, so if you had the possibility to quantify the stress-related hormones in your animals, because some recent papers are telling that even short-term restraint is causing long-lasting changes in the physiology and in the activation patterns that we see with MRI, with functional MRI. So I was wondering if you have the possibility to quantify the stress levels and if it was, if there was a relationship between this.
We have not done that, but there are people in Newcastle, right? Where is Chris? In Newcastle, they have been looking at stress hormones. I'm not entirely sure this was in the context of fMRI, but I think it was definitely in the context of behavior and electrophysiology. Correct me if I'm wrong. And that the problem is not that exaggerated as you may say there. I do have, and it's actually more than anecdotal evidence, however, in quite a few animals that we train. So this, I skipped that part completely. We heavily train the animals before they go to the magnet, because magnet time in our hands is very expensive in terms of finances, but also man hours and so forth and so forth. So we really train the animals as good as it can before they get in there. And we have had a substantial number of animals, basically, that were in the regular setups, not in a scanner, but in the regular training setups, doing okay, but not really great. They go to the scanner and they work much, much better. And I have no, well, this is it's still anecdotal. There might be several factors contributing to that. When you scan for a long time, the temperature goes up a little bit, you have the vibration, the magnetic field. I have no clue whatsoever what is causing it, but we had quite a few animals actually that were doing much better job in the scanner than around. The other way around, sometimes we have animals that have to adapt 1 day or 2 days when they were novel into a scanner environment. But usually within 2-3 days, they get to at least the same performance levels as in regular training setup. So in rodents, that definitely has been tested, and I think it is much more stressful in awake rodents than in monkeys, which have been heavily, and I must say they're very heavily trained. They know day after day after day what to expect, and also in regular training, it happened it happens quite often that these guys, they are housed in very nice housing facilities, that they are very happy to come into a training setup to do for them sort of like all kinds of interesting tasks. And I think Chris may add some words. There's a mic. There's a mic.
Sorry, we need to, I think, go on. We have a projector, so now we're going to go to the final talk by Gustavo Deco, who has asked me to tell you that he's a nice guy and that Barcelona is in Spain, both of which I'm happy to do, and now hand over to you.